All right, I am going to try and do meiosis, this time with just one chromosome. Um, Hopefully, I can get through this without making too many mistakes. So, we are going to start out with sister chromosomes, one from the dad, one from the mom, and I'm going to try and draw a few things while we're going on and doing this. So, we're going to start with interphase. So, I'm going to once again show you my notes as I get going, and we're going to just do the handheld camera here. So here on interphase, the DNA becomes replicated and sister chromatids are joined at the centromeres. So each of these would unzip and be replicated semi-conservatively. So we would have a matching pair, and these noodles keep rolling. Okay. And then once everything's replicated, we have sister chromatids joined at the centromeres, then the DNA starts to hypercondense, and those homologous chromosomes form the pairs. And if we do have crossing over, this is where it occurs. Now, I tried to dye some pasta like halfway pink and halfway blue, and it just bled into each other. So I'm not going to use any crossing over. But this is where we get the genetic shuffle that is so important for um, genetic variation and genetic and evolutionary purposes, this crossing over. And I wish my pasta would have done half and half, but it just bled and made purple pasta instead. So once we get into this prophase and we have the centromeres and formed and the cell membrane starts breaking down, those homologous chromosomes line up along the metaphase plate. So if we have two kinetochores on opposite sides of the cell. So I'm going to just draw in a kinetochore here and a kinetochore here. So this would be in the middle of the cell. We're going to get these lining up in the middle of the cell, and we're going to have different spindle fibers joining at the centromeres. And I didn't draw it to the center, but I should have, to these different chromosomes. Okay? And once we get into anaphase, then these spindle fibers start shortening up, and they start moving these chromosomes to the opposite sides, close to the kinetochores. And once we get those chromosomes at the kinetochores and divided, then we go into telophase 1. Those spindle fibers start to dissolve, and we get two nuclei that are formed around the two new daughter nuclei that are formed. And then once those nuclei are formed, then we start getting the cell wall, if it's a plant cell, to start creating a cell wall along that metaphase plate, or if it's an animal cell, we start getting a nuclear membrane starting to pinch off here, and it pinches off along the metaphase plate to form two daughter cells with the nuclei and the cytoplasm. And that takes us to cytokinesis 1. So once we get those two daughter cells, we can go into prophase 2, where we get the chromosomes condensing again, and we get spindle fibers forming at the kinetochores. So we will have kinetochores. Let's separate these out even more. So we're going to have kinetochores. But this time, the spindle fibers just attach to not the sister chromatids, but the individual chromosomes. So we don't have them necessarily paired up. Okay. The chromosomes line up along the metaphase plate within, but we didn't have a duplication. So we don't have a pair here on prophase 2. Then we get to metaphase 2. The chromosomes finish lining up on that metaphase plate. And once they finish lining up, these spindle fibers start shortening up. And those kinetochores start reaching the chromosomes. And in anaphase, the centromeres of each of those chromosomes split and separate, and these kinetochores start shortening out their spindle fibers once again, and we get the chromosomes to the individual kinetochores, and they separate. In telophase 2, we have haploid nuclei that form. I'm sorry, my paper is... So we get these haploid nuclei that just have DNA for half a cell. They don't have a matching gene or a matching duplicate. They only have one chromosome in each cell for each type of chromosome. And we get these haploid daughter cells. And each haploid, if we had crossing over, can be genetically unique depending on how the crossing over happened. And this is where we get that beautiful shuffle of genetic material for our genetic variety and our evolutionary processes. And then we get cytokinesis happening, where we get a cellular membrane being made, and we have 4 haploid daughter cells at the end of this meiosis process. And these haploid cells are called gametes. Now, if it's done in the male reproductive system, we will call these sperm. And if this is done in the female reproductive system, we would call these eggs. And we'll see you later. Bye.